Hello to everybody. Let's discuss chapter number 2. Okay, let me go into the slideshow mode. Okay. So in chapter number 2, we are going to talk about the tools of the laboratory. So what are the tools available in a microbiology laboratory? Okay. So basically, the aim of the microbiology lab is to identify the bacteria. So what kind of bacteria is there in the patient sample, or what kind of bacteria is responsible for causing the infection. So for doing that, we need to follow 5 I's. Now what are those 5 I's of microbiology? Inoculation, incubation, isolation, inspection, and identification. Okay. So first of all, what is inoculation? So basically, the sample is taken, and then it is basically that sample is inoculated, or that sample is introduced into the growth medium. Okay, I will explain you what growth medium is also. Okay, so and then that sample which is placed on a growth medium, it is then incubated. So when the sample is incubated, which means so in the sample the microorganisms are there. So that sample can be a blood sample, it can be urine sample, sputum sample, semen sample, or whatever the sample is. So first we need to add that sample, or we call it an inoculum, to the medium. And what is medium? Medium is basically medium basically contains the nutrition for the growth of the microorganisms. And then that medium with the sample is incubated. So in the microbiology labs we have incubators, and those incubators are maintained at a specific temperature. So most of the microorganisms, they grow at 37 degrees Celsius. So that plate or the liquid broth, I'm going to discuss with all very shortly, the different kinds of mediums. They are placed in the incubator, which is maintained at a specific temperature. Mostly we use 37, and some of the organisms, some of the microorganisms also need specific gases for their growth. So everything, all that requirement is there in the incubator, and that the requirement is for the growth of the microorganisms. So that is what incubation means. And once we do the incubation, then we isolate the bacteria. Sometimes when you have isolated some culture from the sample, sometimes it's a mixed culture, then we have to inoculate it and re-incubate it, and then we can get the separate pure cultures. And then we have to inspect the cultures. We can do it microscopically or macroscopically. Like if you're like you can see here, this is a petri dish in which some bacteria is growing in the form of some colonies. You can see them with your eyes, or you can also watch. You can also take some bacteria and make a kind of a smear and stain the cells, stain the microorganisms, and you can see under the microscope. That's basically the inspection. And identification. So basically, identification can be done by genetic methods. You can isolate the DNA, and you can run various kinds of gels, the different kinds of genetic methods, and by which we can finally identify the microorganisms. So these are the 5 I's of microbiology: inoculation, incubation, isolation, inspection, and identification. Okay. Now, before we understand all these steps in detail, you need to know a few terms. So first of all, what is culture? You'll be coming across this word culture maybe every session. Culture. So culturing means to grow the microorganisms. Okay, whenever the word comes culture, it means to grow the microorganisms. And now how to grow the microorganisms? So the microorganisms can be grown in some medium. Now what is medium? Medium has the nutrients for the growth of the microbes. Like this is one of the characteristic of life forms, that we all need nutrition for growth, right? We eat food 3 times a day. Why we are doing it? So that we can nourish our body. So the food we eat gives us energy. So similarly, microorganisms, they are very small, but they are form of life. So they also need nutrients for the growth. and that nutrition is present in the medium. Okay, then inoculum is a small sample of the microbes. So this is just a sample of the microbes. Inoculation means the introduction of the inoculum into the medium to culture the microbes. So now all the three terms which we have learned here, they are all being used in this definition. So the introduction of the inoculum, what is inoculum? A small sample of the microbes into the medium. So what is the medium? It's a nutrition for the growth of the microorganisms to culture the microbes, which means to grow the microorganisms. Now, as I said, the clinical samples like the inoculum, which is a sample of microbes, it can be from your body fluids, discharges, anatomical sites, or diseased tissue. Okay, it can be a blood sample, urine sample, skin swab, sputum. Any specimen can be there. Any specimen can act as your inoculum. Now, as I said, next step is incubation. So we do the incubation in the incubators, and these incubators, they maintain specific temperature. The organisms, this is a range of the temperature the microorganisms need. They usually grow between the range of 20 to 45 degrees Celsius, but majority of the microorganisms, they grow at 37 degrees Celsius. And these microorganisms, they can also have a requirement for some gases like oxygen and carbon dioxide. So all these requirements are met in the incubator. So in the incubator, all these conditions are there which are good for the growth of the microorganisms. So now you can see here, these are the cultures. Okay, so this is a kind of we call them slabs. So this is a petri dish, and in the petri dish, the medium is there for the growth of the microorganisms. Okay, so the microorganisms can be grown in these petri dishes, or they can be also grown in these kind of tubes. And the medium can be a liquid medium, it can be semi-solid medium, or it can be a liquid medium. Okay, so these are called slants. So here the medium is little slanting, and this is a petri dish in which the medium is poured. I will tell you what material is there, and it becomes a solid. Okay, so this is showing you pure culture, which means you can see all this whitish streak you see, or this yellowish streaks you see here, or this, in this case, the reddish. The different microorganisms, the different bacteria. Okay, so in this white streak you see, it has one kind of a bacteria. This is yellow, this is one kind of bacteria. This is red, this is another kind of a bacteria. So this is called pure culture. Okay, when there is only one kind of a microorganism, then it is a pure culture. And if you see this culture, you can see like these are called colonies. Okay, every colony represents one bacterial cell. Okay, so in this you can see there are some colonies which are white in color and some are yellow in color. So this is called a mixed culture. This is not a pure culture, unlike like these. Okay, so here all the bacteria is of one kind, in this all the bacteria is of one kind. But if in this plate you see there are white colonies, there are yellow colonies. So these are two different kinds of microorganisms, two different kinds of bacteria. So this is called a mixed culture. And if you see this plate, in this plate you can see there are some red colonies, and majority of the colonies are red, and at only one place if you see there is some kind of a whitish thing growing. Okay, so this is not a mixed culture, but this is called a contaminated culture. Why it's called contaminated culture? Because the original culture is red, the colonies are of red color, which they are representing one kind of a bacteria. But in this pure culture is growing something else which the microbiologist has not grown. Okay, so this is coming from atmosphere. So there is something wrong, some error while they were trying to grow this bacteria, that something has come from the atmosphere. So this is a contaminated culture. So I hope you understand what a pure culture is, what a mixed culture is, and what a contaminated culture is. Contaminated culture is not a good culture. If your plates are getting contaminated, which means that you are not culturing the microorganism in a perfect way, because microorganisms, as we learned in the last lecture, they're ubiquitous, they are everywhere. So it means that while you were trying to culture the bacteria, there was something wrong in your protocol, or something, you did something wrong while performing the culture, that something from the atmosphere is growing in your plate. Okay, now the culture medium, as you just talked about, medium is that which provides a nutrient for the growth of microorganisms. Now this culture medium, it can be in the test tubes, it can be in the flasks, it can also be in the petri dishes. So as I just showed you, this is a petri dish, these are the tubes. Okay, so we can, it's just a medium, and that medium can be poured in petri dishes, flasks, or the test tubes. Now for inoculating the medium, what we have to inoculate, inoculum. And what is inoculum? It's your sample, and it can be sample from your clinical samples, or it can be any kind of a sample. If you want to grow some bacteria from your water, then that's going to be your inoculum. So it all depends what kind of experiment you are doing. Now how to do the inoculation? It can be done with the help of loops, needles, pipettes, or swabs. Okay, so if you are taking the laboratory course also, you will be learning all these things, how to inoculate the medium. We inoculate the sample on the medium using loops, needles, pipettes, and the swabs. Okay, now the medium, as I said, it can be liquid, it can be semi-solid, or it can be solid. And some of the solids can be converted into liquids, and some of the solids can be such which cannot be liquefied. And mostly when we have the medium which is solid in nature, in that we add a polysaccharide which is isolated from gelidium, which is a kind of a algae, and this polysaccharide is called agar. Okay, and the property of this agar is it is solid at room temperature, and if you heat it, it is going to liquefy. Okay, so if you warm agar, it is going to become liquid, but at room temperature it is solid. So agar is a kind of a complex polysaccharide, very important component of the solid mediums. So you can see here, this is a liquid medium, this is semi-solid, and this is the solid. So it all depends what kind of microorganisms you're growing, or what is the purpose of your experiment. On the basis of that, you can use a liquid medium, you can use semi-solid medium, or you can use solid medium, and all these things will be more clear to you in the lab. Okay, now we're going to discuss some different kinds of the medium. So because in microbiology, the medium is very important. So medium is the nutrient for the growth of the microorganisms. So there are different kinds of mediums for the growth of the microorganisms. So the medium can be a defined or synthetic medium. So when we use a defined medium, so what does the word defined means? This is a kind of a medium whose composition is precisely chemically defined, which means that we exactly know what is the amount of different kinds of amino acids are there in the medium, or how much is the sugar in the medium. So we very well know that what are the components of that medium. Let me give you an example here. Like on the next slide, if you see, this is a defined medium for the growth and maintenance of Staphylococcus aureus. So this is a defined medium. So you can see that we know that this medium has 0.25 grams of each of these amino acids: cysteine, histidine, leucine, phenylalanine. I don't know if you are aware of what amino acids are. Amino acids are the subunit of the proteins. And in this medium, we have some of these amino acids, and we know very specifically that their amount is 0.5 grams and 0.12 grams of these specific amino acids. And there are some other constituents in this medium, which is for the growth of this bacteria called Staphylococcus aureus. We know exactly how much each component is there in the medium. So these kind of mediums are called defined mediums. Then we have another kind of mediums which are called complex mediums. So in the complex mediums, one or more component is not chemically defined. So why it's not chemically defined? Because these kind of mediums, they contain extracts of some animals, plants, or the yeast. Okay, some of the mediums, they have blood in them, or serum in them, meat extracts, milk, yeast extracts, soybean digest, and peptone. So if the medium has blood or serum, so they are going to act as source of nutrients, but we do not know exactly what is exact content of that specific amino acid or that sugar. So those kind of mediums are called complex mediums. So you can see here, this is another kind of medium for the growth of same bacteria, Staphylococcus aureus, and it is called brain heart infusion broth. Okay, this is a complex medium for the growth and maintenance of Staphylococcus aureus. So this is, which we just discussed, this is this is the defined medium for Staph aureus. This is the complex medium for Staph aureus. So let's see what components are there. It has 27.5 grams of brain heart extract and peptone extract. So we do not know exactly what is what in the brain extract or heart. It's definitely going to act as a source of nutrients, but we do not know exactly which amino acid is in how many grams and all that. So these kind of mediums, they're called complex mediums. Okay, we do know it has 2 grams of glucose, 5 grams of sodium chloride, but because of the presence of the heart extract or peptone extract, we do not know exactly what constituents, in what exact amount are there in these heart and the brain extracts. So these kind of mediums are called complex mediums. I hope you understand what's the difference between a defined medium and a complex medium. Now we are going to come across more terms. It's very important in microbiology to know about the different kinds of mediums, because mediums are the source of nutrition, and on the basis of the nutrition which is there for the growth of the microorganisms, we grow or we culture different kind of microorganisms. So we have some mediums called general purpose mediums, so which means that they are generally complex mediums, which means they have good nutrition, but we are not sure exactly what is exactly the amounts of each component. And general purpose mediums, they have good nutrition, and they are like they can on these kind of mediums a broad spectrum of microorganisms can grow. So they're very rich in nutrition. So most of the microorganisms, they can grow on these kind of general purpose mediums. Now the enriched mediums, next term we need to know is enriched medium. So they contain complex organic substances such as blood, serum, hemoglobin, or some specific growth factors for the growth of some fastidious microbes. Fastidious microbes are those microbes which are like very stringent in their requirements, that they will grow only if this is there or this component is there. So that is the purpose of using the enriched medium. Enriched medium contains quite a good kind of a nutrition, because they contain complex organic substances, so they can serve as a good nutrition for the growth of some hard to grow microbes. And this kind of medium can also encourage the growth of some of the bacteria which are there in the low numbers, because it's a good nutrition. Okay, so I hope you understand what are general purpose medium and what are enriched mediums. So this is blood agar, this is chocolate agar. So these are examples of enriched medium, which means we can grow some of the fastidious, some hard to grow microbes on these kind of mediums. Now very important medium is a selective medium. Now what is a selective medium? Selective word means something which selects. So these kind of mediums, they basically select for the microbes. They do not let every organism to grow on them. So they have something in them, they have some components in them, some agents in them, which are going to inhibit the growth of one kind of a microbe, and they will let the other kind of microbe to grow. So some of the mediums, like they can let gram, I'm going to talk about what are gram positive and gram negative bacteria. Some of the medium can let gram positive bacteria to grow on them, but they will inhibit the growth of gram negative bacteria. In some of the medium, they may have something in them, some agent in them, which will inhibit the growth of gram negative bacteria on them, and they will select for the gram positive bacteria on them. So that is what is the meaning of a selective medium. So they have something in them that selects for the growth of a specific kind of microorganisms. Okay, so sometimes like when you are having some sample from some clinical, you have some clinical sample now, or suppose you have some skin swab. Now on the skin also a lot of different kinds of bacteria are growing, but you want a specific kind of a bacteria to grow on them. Then you take the sample and you put them and you inoculate them on a selective medium. I mean, it will not let all the different kinds of microorganisms to grow on this kind of a medium. It will select for the kind of the microorganisms which are going to grow on them. Okay, differential medium. Now differential medium is that medium which will not select for a specific kind of microorganisms. It will let all the kinds of microorganisms or multiple types of microorganisms to grow on them, but the multiple types of microorganisms which will grow on this kind of the medium, they will be showing different characteristics, and those different characteristics can be in terms of color or different colony size, maybe production of some gas bubbles. So basically differential medium can let multiple types of microorganisms to grow on them, but while growing on them they will show different characteristics, maybe in terms of the different size of the colonies or different colors of the colonies, different texture of the colonies, or maybe the color of the medium changes, so that a microbiologist can actually tell, looking at the plate, this is this organism, this is this organism. Like if they are showing pink color, they are fermenting lactose, or if they're showing white color, they're not fermenting lactose. So that is the meaning of a differential medium. So it lets multiple microorganisms grow, but those multiple microorganisms growing on this kind of medium, they will show different characters, and a microbiologist will be able to differentiate them on the basis of their different characters. Now you can see here, this is very good picture to understand the difference of selective and differential medium. You can see here, this is a mixed sample, and if you take the inoculum from here and put on the plate, you can see that this is a general purpose medium which will let everything to grow, because it's a general purpose, broad spectrum. It will choose all the microorganisms. You can see all of them are growing here, but on this plate you can see that only these big colonies are able to grow, because this is selective medium. It is not letting some of the kind of, so here actually you see like some are little dark colonies, some are white. So this selective medium is letting only white colonies to grow. Now it's the same thing here, mixed sample. This is your general purpose medium, and you can see that your gray and the white colonies are growing. But on the differential medium, if you take this culture and put the differential medium, you can see differential medium is letting all these kind of microorganisms to grow, but you can see that they are showing different colors. So some are blue, some are white, some are red. So on the basis of the color, we can identify that this is this kind of a bacteria, this is this kind of a bacteria, this is different, this kind of a bacteria. So that is the meaning of a differential medium. Okay, here's a very good example. There is one agar, one medium which is called McConkey agar. Okay, this is a very good example that this medium acts both as a selective and differential medium, which means it can select a specific kind of microorganisms, and the microorganisms which this medium selects, it can also differentiate between the ones which are growing on them. Like McConkey agar is a example of selective and the differential medium. So basically McConkey agar does not let the gram-positive bacteria grow on them. It's the gram-negative bacteria which will grow on them, and then among the gram-negative bacteria which are growing on them, you can see that these are all gram-negative bacteria growing on them, because gram-positive bacteria cannot grow on them. So out of those gram-negative bacteria which are growing on the McConkey agar, some bacteria can be fermenting lactose. Now what is lactose? It's a kind of a sugar. So some of the bacteria can be fermenting lactose, and some of them may not be fermenting lactose. So in this medium there are added some pH indicators. So the microorganisms which can ferment lactose, they are going to form red colonies. Okay, and the bacteria which do not ferment lactose, they're going to show white colonies. So there are pH indicators in this medium which are going to change their color if the bacteria is going to ferment the lactose. If it is a lactose fermenter, the colony will be red in color. If the bacteria is white in color or yellowish in color, it is not a lactose fermenter. Okay, now there are some more terms. Reducing medium. Now what is this reducing medium? It's a kind of a medium. It contains a substance that can absorb oxygen or slows the penetration of oxygen. So this kind of a medium is important for the growth of anaerobic bacteria. So we will talk more about what is the aerobic bacteria, what is the anaerobic bacteria. Aerobic bacteria are those bacteria which require oxygen for the growth, and anaerobic bacteria are those bacteria which require oxygen for their growth. So reducing medium is for the growth of anaerobic bacteria. Now transport medium, usually sometimes the clinical sample is taken from some hospital or that, so then that specimen has to be transferred to the lab where the specimen is going to be cultured. The bacteria which is there in that sample, it is going to be cultured. So it means that the specimen has to be transported from the hospital to the lab. So sometimes because the bacteria can die if we do not take care of these bacteria in these specimens, so they have to be transported in a medium which is called a transport medium. So it's used to maintain and preserve specimen that have to be held for a period of time before clinical analysis can take place, and that's going to take place in the lab. Okay, another kind of a medium is carbohydrate fermentation medium. So these medium basically contain a sugar in them which can be fermented, and these medium also have a pH indicator to show the reaction. So you can see here, like this is a control. Okay, if the pH of the so these mediums, they have sugar in them. Okay, and if it is, the sugar is fermented, after that the pH, if the pH is alkaline, then this is the color which will be shown. If the if the pH becomes acidic, then this medium turns yellow in color. And also in this medium you can see there is a tube here, and if there is a production of gas during the process of fermentation, and there is a formation of gas during fermentation, so there will be the entrapment of some bubbles here. Okay, so that can show us if the gas is also being produced during the fermentation process. So this kind of a medium contains some sugars in them with the pH indicators. So if the pH is alkaline, this is the color. If it is acid, yellow is the color. And if with acid there is a gas, you will also see the color will be yellow, and there will be also formation of some bubbles. Okay, then there's another kind of a medium called assay medium. Now assay mediums are used by the microbiologists to test the effectiveness of some antimicrobial drugs. Okay, so basically these kind of mediums are used to check the effectiveness of some disinfectants, antiseptics, cosmetics, and all that. Enumeration medium, these are the mediums which are used to count how many microorganisms are there in milk, water, food, soil, or other samples. So if you have all these samples and you want to know how many microorganisms are there in these samples, we use a medium called enumeration medium. Okay, so we just discussed about various kinds of mediums which are there. Now we are going to talk about medium is used for culturing. So after you add your specimen, or you just put your inoculum on the medium, you incubate it, and then after that we incubate it for specific time period, and then we isolate the microorganisms. So how do microorganisms look on the medium after they are incubated? If you will look at the plates, you will see the colonies. So let me show you some colonies here. Okay, these are all colonies. So the microorganisms on the plates, they grow as like these colonies, like these. If you see that microorganisms in the petri dishes on the solid medium, they grow as colonies like these. and every colony every colony has millions of bacterial cells in them, and every colony has originated from one single bacterial cell. Okay, so every colony represents one bacterial cell. Okay, so from one bacteria it forms 2, 2 to 4, 8, 16, 32. Okay, so it keeps on multiplying and forms this kind of a colony which can be seen with the naked eyes. Okay, so every colony has originated from one bacterial cell, and every colony represents one bacterial cell. But every colony actually has millions of bacterial cells. Because it has originated from one single cell, we consider it as one cell. So what is a colony? A macroscopic cluster of cells appearing on a solid medium arising from the multiplication of a single cell. And for isolation, we need a medium with a firm surface, like I just showed you, a petri dish and an inoculating loop. So all these things we will be doing in the lab part. So here it's very nicely shown that if you have a mixed culture like this, you can see some bacterial cells are red in color, some are white. This is just to represent you different kinds of cells, so you can separate them. Okay, so they will be showing you as a separate colonies. You can separate them, and you can put them on the medium, and you can see how they are showing different colonies. So if this is your mixed culture, okay, if this is your mixed culture, this is one kind of bacteria, this is another kind of bacteria, and if you can separate them, these are two different kinds of bacteria, and you can put them you can separate them, and you can see on the petri dish they are forming different colonies. Okay, so this is your mixed culture. This has two different kinds of bacteria, and when you take these bacteria and put them on the petri dish, they are forming different colonies. Okay, so this kind of bacteria, they're forming this kind of colonies, and this kind of bacteria, they're forming this kind of colonies. It's basically just to show you that how the mixed population of the bacterial cells, they're going to form different kind of colonies on the petri dish, and you can see these colonies with your eyes. Okay, how we can inspect and identify these microorganisms? So once you have the microorganisms growing on the petri dishes, we can use microscopes to see them. Okay, or we can also do various biochemical tests, because all these different kinds of bacteria, they have different enzymes in them. So by doing various biochemical tests, we can detect what kind of enzymes are present in them, and on the basis of that we can identify the bacteria. We can also do various immunological tests and the genetic tests. Okay, so we follow a approach. So first of all, we can see them. First of all, we can look at the colonies, which we can just see with our eyes. Then we can use a microscope. We can take these bacterial cells, we can make a kind of a smear, we can stain them, see under microscope. We can also look for various enzymes present in them. That is done by biochemical test. We can do immunological tests. We can also do genetic tests, and that's the way we identify the microorganisms in the microbiology lab. So for checking the for checking or for doing the microscopic analysis of the microorganisms, we use this kind of a compound light microscope, which we are going to teach you in the first lab, how to use this compound microscope. Okay, preparing the specimen for the microscope. Okay, so this is a microscope which we're going to use, but how to prepare the specimen, and this is the stage. Okay, usually the sample which is taken, the bacteria are growing on the petri dishes. We can take a few cells from the colony and we can make a smear on a slide. Okay, and then we can stain them and put a cover slip, and the slide is usually placed here on the stage. Here is the condenser lens and these are the objective lens. So this is the stage, the sample of the microorganism which is now on the slide. The slide is placed here on this stage. Okay, now it depends, as I just said, that we can take the bacteria and we can stain them. When we stain them, the bacteria usually dies. Sometimes there's a requirement when we want to see the microorganisms in a viable form, which means in the living form. Then we have to do a hanging drop technique, or another way is to just make a wet mount. Okay, so how the specimen is prepared, it depends on the condition of the specimen. We want to see them, is it okay to see them just dead, or we have the requirement of the experiment that we want to see them in a viable state, or how many details you want to see of the microorganism. You want to see the structure, you want to see the movement. It all depends. So if you want to see the cell in a viable form, do wet mounting. So what is basically wet mounting? We just take the drop of the specimen, or we can have take one or two drops of the culture and put the specimen in them, and just put a cover slip and just watch them under the microscope. So suppose you want to see, if you have a pond water and you want to see what different kinds of microorganisms are there in the pond water. Suppose you want to see that. So you can just take a drop of a pond water, put it on the slide, cover it with a cover slip, and watch under the microscope. That is called wet mounting. Or another way is called a hanging drop method or hanging drop technique. So for this, basically we use a special kind of slides, and these are called depression slides. So this is a slide. It doesn't have a smooth surface. It has a kind of a cavity here, or it's called a depression cavity. And here, basically in this depression cavity, you can just add your sample, and then you can have a cover slip, and you can also put a little Vaseline here on the cover slip, so that all this side and this side gets sealed. And then this is a special kind of a slides. These are called depression slides, and these are used for hanging drop technique. So you can also see the specimen in a viable or in a living form by using this kind of a technique. So it's nothing great. It's just a different kind of a slide in which there is a depression cavity, and this slide is called a depression slide. And you can just put your sample in this depression cavity, and then just cover it with a cover slip and just watch under the microscope. Now, if you want to see a lot of details of the microorganism, then I mean, if you basically don't stain the samples, it's very difficult to see them. So you can stain the microorganisms, then you can see them more clearly and you can see more details. And for staining of the microorganisms, we use different kinds of dyes. So we have different kinds of dyes: the basic dyes, the acidic dyes. So basic dyes are the dyes which have a positive charge on them, and acidic dyes are those dyes which have negative charge on them. So usually the bacteria, they have numerous negatively charged substances. So so if they have negatively charged substances, which means that those negatively charged substances and the bacteria, they are going to attract the basic dyes. So which means that basic dyes can go and bind on the bacteria. But the acidic dyes, because bacteria have negatively charged substances in them, acidic dyes, they are going to be repelled by them. Okay, so these are two kinds of stains. So we have two kinds of stainings, positive and the negative stainings. So what is the positive staining? So it's a kind of a staining in which the dye is going to stick to the specimen and gives its color. Okay, if the dye is going to go and stick to the specimen, it's going to give its color, right? That kind of staining is called positive staining. Okay, and the positive staining can be done with basic dyes because they have positive charge on them and they can go and stick to the bacteria. So that's a positive staining. Now what is negative staining? So negative staining is that kind of a staining in which you are using a dye which does not stick to the specimen. And what does it do? It just settles some distance from its outer boundary. Okay, and we use these two kinds of, so basically negative stain does not stick to the specimen, it just sits around the outer boundary of the microorganism. So there are these two kinds of dyes which are used for negative staining: India ink and nigrosin. Okay, now we need to know more about the stainings. So we can use some simple stains. So simple stains are those stains which only require a single dye, and it's an easy process, and it can basically when we do the simple staining, so all the cells in the smear, because when you take a bacterial sample and you make a smear, there are various cells in the smear. So when we use the simple stains which involves a single dye, it's going to go and stain all the cells. Okay, so causes all the cells in the smear to appear more or less the same color, because it is going to go and stain all the cells in the smear. Okay, differential stains, basically they involve two different kinds of dyes. They have a primary dye and then they have a counter stain. So differential stains, as again the word says differential, it can differentiate between different kinds of cell types or cells. Okay, one of the very typical example of differential stain is gram stain, which I'm going to discuss. First of all, let's see what the simple stains are. So simple stains, as I just said, they require only one single dye. So here is crystal violet of Escherichia coli. So Escherichia coli, E. coli, which we discussed in chapter number 1. So you can see that how the crystal violet stain is staining all the cells in the same way. And this is a methylene blue of Saccharomyces. So you can see this is Saccharomyces, how they are getting stained in the same way with this simple stain, which involves just one dye, methylene blue. Okay, gram stain. This is a very typical example of a differential stain. Okay, this is gram staining is very important technique, and we will be learning in the lab also. This was developed by a scientist called Christian Graham. So this staining involves basically two dyes, and it also involves one mordant and also decolorizer. So there are basically 4 steps in this kind of staining. So it involves crystal violet, grams iodine, alcohol, and safranin, which is a counter stain. I'm not going to discuss the protocol here because that's a part of the lab. So basically, gram staining can differentiate between the different kinds of microorganisms on the basis of the kind of cell wall they have. Okay, so it can the gram positive bacteria, they have a thick cell wall, and gram negative bacteria have this they have a thin cell wall. So gram positive bacteria, they look violet in color, and they are called gram positive bacteria. And gram negative bacteria, because they have a very thin cell wall, they are not able to retain the first dye, which is the crystal violet. So the crystal violet is lost by them, and then they are stained by the counter stain, which is the safranin. So they appear pink in color. Okay, so the gram negative bacteria, by this kind of staining, they appear as pink in color. So gram positive bacteria, because they have thick cell wall, they're able to retain crystal violet dye, which is the first dye in the procedure. And then because they're thick, the thick cell wall can retain crystal violet color, so they look, after staining, they look violet in color. So they are gram positive. Gram negative bacteria, because they have thin cell wall, they're not able to retain crystal violet dye. So the crystal violet dye is lost, and then they are stained with the counter stain, which is safranin, and the color of safranin is pink. So the gram negative bacteria, they appear as pink in color. Okay, then the another kind of differential stain is acid fast stain. Okay, the acid fast stain basically differentiates between acid fast bacteria. So fast word comes from resistant, and the acid fast bacteria, they appear as pink in color, and the non-acid fast bacteria, they appear blue in color. Now acid fast bacteria, they have a very impermeable cell wall, and the dye cannot enter inside the bacteria because their cell wall is very impermeable. So the dye is basically binds outside the cell wall. So that is why they are called acid fast bacteria, and they appear, because the dye all binds outside the bacteria, they are pink in color. Okay, so these bacteria cell walls have a particularly impermeable cell wall that holds fast tightly to the dye, and the dye is carbolfuscin, which is pink in color. Okay, so this dye sticks to the cell wall. So that is why outside the cell wall, that's why they appear as pink in color. And the other kind of bacteria, which are non-acid fast bacteria, they appear blue in color. And this was a method which was used to detect Mycobacterium tuberculosis. This is a bacterium which causes tuberculosis. And the another kind of differential stain is called endospore stain. Um, this is basically a staining method which is very similar to acid fast staining, um, because in these this is a method of staining for staining the endospores, and in this kind of a staining the dye is forced by heat to enter into the resistant bodies called endospores. So spores are very resistant bodies. Okay, so the dye is forced by heat to enter into these endospores, and this kind of a staining is can distinguish between endospores and the vegetative cells. Okay, and it is can identify the gram positive spore forming members of the genus Bacillus and Clostridium. So this picture shows you all the types of differential stains I just talked about. So this is gram stains. You can see all the bacteria which are showing which are purple in color or violet in color, they are gram positive, and all of the pink ones are gram negative. And here the reddish purple ones, all these reddish purple ones, they are acid fast, and the blue ones are non-acid fast. Okay, and this is the endospore staining. In this, the green ones are the endospores, and the vegetative cells, they are stained as pink. So by doing all these three different kinds of stains, we can differentiate between the different kinds of microorganisms. And in the endospore staining, we can differentiate between endospores and the vegetative cells. Okay, then we have some special kind of stains for staining capsules, and capsule is the outer surrounding outside some of the bacteria. India ink dye is used to stain the capsules, and then we can there's another staining which is called flagellar staining. Now what are flagella? Flagellum are basically long appendages in some of the bacteria which is required for the locomotion. So we can also see the flagellum also. And this is the capsular staining. Okay, India ink dye is used to do the capsular staining. It's kind of a microorganism. Its name is Cryptococcus neoformans. So you can see this is outer covering around this bacteria. This is called a capsule. Okay, and with this, it's the staining is done with the India ink. And all these bacteria, you can see this is called Proteus vulgaris. You can see this, all these appendages you see here, they are flagella. So this is a flagella kind of a bacteria. So these are some of the specific stains. So this basically finishes this talking about lab. I hope you can always contact me. Thank you much. And this finishes this chapter.